Hello, my name is Daniele Bizzarri. I am a PhD candidate in the group of molecular epidemiology in the LUMC in Leiden. And today I would like to talk to you about my research, which is called H1NMR metabolomic surrogates to impute common clinical risk factors and endpoints. So what is done in epidemiological studies is to collect samples for a large amount of individuals. So we collect samples and then we calculate the levels of biomarkers and biological information that can help us to understand the health and to distinguish the unhealthy and healthy individuals. So we have large amount of data, but we still need methods to understand the different phenotypes of these people. And what we like to do, for instance, is to use simple linear models, simple statistical learning to understand this information. And for instance, is famous the work of Steve Horvath, which is able to calculate aging using simply DNA methylation. And he predicted very well. But in our group, what we like to use is metabolomics. This is because it's an omics that is very much related to the genetics of the individual, but also to the environmental factors that can affect the individuals. So my professor, Eric van den Akker, was able to create a model that can very accurately predict aging, while Joris Dalen, our colleague, was able to use the same platform to predict mortality. And he predicted very well, even better than a set of clinically used risk factors. So we wanted to extend this research line and try to see if we can predict more variables. For instance, can we predict obesity or medication usage or even diabetes status? So we try to do this with the platform called Nightingale Health. So it's a Finnish company that can calculate the measures of the metabolites of 226 features just using 100 microliters of blood or other sample types. And this is very standardized and simple techniques. Therefore, it's used in epidemiological studies in very large consortium, for instance, the UK Biobank, but also BMRI-NL, which is our consortium. BMRI-NL is a consortium composed by 26 Dutch cohorts and comprise the metabolic measurements of 26,000 individuals, which is a very big amount. And here you can see the age distribution of these individuals, and you can see that it goes from 18 years old to 100 and plus years old. So it's a very broad age range. So we have all types of individuals, and therefore our models can learn from all these different metabolic profile to distinguish different type of health. So the models are logistic and estimate regressions. And they are trained with an alpha equal to 0.5 and we tune the lambdas. We use two evaluation techniques. One is a double five-fold cross-validation consisting of dividing the dataset in 5 folds and training the dataset on 4 of the 5 folds and then testing the dataset on the remaining folds, doing this 5 times and obtaining 1 accuracy score. But also a Live1 biobank out validation, which is done doing similar things, but in this case, leaving out an entire biobank. Both of them internally have an inner loop, which is a five-fold cross-validation and is used to tune the hyperparameters. So in particular, we like to use also the Live1 biobank out validation because it is an estimation of what would happen if an external cohort would like to use our models. However, we think that the double five-fold cross-validation is the more unbiased evaluation. Therefore, that's what we use to evaluate better our models. And here is the results of the double five-fold cross-validation, which is, you can see the results, the accuracy results in the color-coded. So you see that we trained 20 different models, all on binary variables. If we had a continuous variable, we binarized it. So we didn't use BMI, but we used obesity level. And you see that there are many types of variables. And these are, for instance, demographic variable, body composition, or clinical endpoints that can be used to evaluate future events as well. So all our models perform quite well, apart one, which is the one for eye blood pressure, and therefore we didn't use it for further evaluation. So once we established that our models were very good, we also wanted to apply them. And one of the very important application we liked was to use them to substitute missing information in association studies. We know that association studies are often hampered by the fact that there are many missing information. Therefore, their statistical power is very much lowered. However, we could use omics dataset to predict the information that is missing, for instance, using our surrogates. What we did here, actually what our collaborator Anna did in this 4 course, she did a meta-analysis to compare a TWAS made comparing the reported variable and the metabolic-based surrogate as a phenotype. So a TWAS is a transcription-wide association studies, which is similar to a GWAS, but in this case using gene expression and compared and associating gene expression to a phenotype, which in this case is the reported variable or the metabolic surrogate. So what she found out is that actually the same gene were associated with the reported variable or the surrogate. And then she did an enrichment analysis, which is what you see here, using GCA and also and using Reactome as a database. And what you see here are the number of pathways that are in common between the two association techniques. So here in blue, and you can see actually that the results are pretty similar. And the most similar ones are in particular the results for the surrogate triglyceride levels or the one for HDL cholesterol levels. So what we did also is to try to study if our models can be used as biomarkers for future endpoints, in particular all-cause mortality. We used the dataset of LLS siblings, which is older individuals, because they were enriched for dying individuals. And what we found out is that a lot of our variables can actually associate to mortality. In particular, 13 of them were associating individually to mortality. And then we tried to do also multivariate analysis to see if they can independently relate to mortality. We did it sex stratified because we know that there are many differences between men and women and old age. And we indeed find out two completely different models, but still with many variables in men that can recapitulate the association to mortality. While in women, only 3 of our surrogates were associated to mortality, independently at least. So, and then we did a simple literature review, and we find out that actually this association were found out also in the reported variable, and then and therefore we think that these models can be very much predictive of future events and future mortality in particular. So after this, we create also an application which is called Mimir, and you can find it on GitHub, but also on CRAN. And this can be used to predict, to impute all these models that I just mentioned, so the 19 surrogates, or also the metabolic age and mortality score, and so on. And also you can use it to explore the differences between your dataset and the BMRI-NL dataset, or to evaluate the accuracy of the pre-trained metabolomics-based score on your dataset, calibrate the scores, but also associate with incident and prevalent events. So in conclusion, we have 19 models that can accurately predict generally measured epidemiological variables. These variables can be indicative for the mortality of these individuals, and we train 2 sex-stratified Cox regression analysis at older ages. This marker can be used to impute missing information in association studies, and then we have an application that can be used to impute these variables on your dataset. So you can find here on this QR code all the publication related to this if you want more information about this. So I want to thank my professors, in particular Eline Slagbone, Marcel Reines, and Eric van den Akker for this work, and also our collaborator Anna for making the TWAS analysis. And thank you for your attention.